Hello my dear students, have you ever thought how plants drink water and get their nutrients from the soil? Plants don't have a mouth like us, but their roots do the job. In this chapter, we will learn how roots take in water and minerals from the soil and send them all the way up to the level. Let's find out how roots keep the plant alive and growing. This is a syllabus overview. The syllabus includes process involved in absorption by roots and movement of water up to the xylem. Now we study what is plant physiology. It's a branch of biology that studies how plants live and function. It explains how cells, tissues and organs of plants work simultaneously. This chapter focuses on osmosis and other essential processes that help plants survive. Absorption by roots. Roots not only support the plant in soil but also absorb water and minerals from the soil. These nutrients are sent up through the stem to the leaves, flowers and fruits. Why do plants need water and minerals? Water helps in photosynthesis. Water is used in green leaves to make food or glucose. Transpiration, water evaporates from leaves as vapor. It helps in cooling the plant and pulling more water upwards. Transportation, water helps in moving minerals from roots to leaves and sugars from leaves to other parts. Mechanical stiffness, water keeps plant cells full and firm that starch it, helps plant stand upright. Minerals in soil, plants absorb mineral salts from the soil as salts like nitrates, phosphates and sulfates, as ions like potassium, calcium, magnesium and chloride. These minerals are needed to make cell parts, build enzymes, support overall plant growth. Characteristics of roots for absorbing water. The ability of roots to absorb water efficiently depends on three key features. Large surface area of roots. Roots have numerous rootlets and root hair which increase the surface area. Even a small plant like balsam has hundreds of root hairs per rootlet. All root hairs laid end to end would cover kilometers in length. More surface area is equivalent to more water absorption. A freshly germinated seed showing the single root with root hairs. The image is showing here. Interesting fact, botanist H.J. Dittmer found that a 4-month-old rye plant had 600 km of total root length, over 14 billion roots here, total root hair length over 10,000 km. This gives a huge surface area to absorb water. Root hairs have cell sap with higher concentration. Root hairs contain cell sap, a watery solution with salts. This sap has higher concentration than the water in the surrounding soil. Due to this difference, water moves into the root hairs by osmosis. This concentration gradient helps pull water in. Root hairs have thin walls. Each root hair has a thin cell wall and a cell membrane. The cell wall is thin and permeable, allows easy movement of water and substances. The cell membrane is semi-permeable. Water can pass through. Larger molecules like salts cannot. These features helps in selective absorption of water. This diagram of a single root hair showing its different parts. Absorption and conduction of water and minerals. The whole process of how roots absorb water and minerals from the soil, move them through the root and send them up the stems happens through 5 main steps: imbibition, diffusion, osmosis, active transport and turgidity and flaccidity. Imbibition. What is imbibition? Imbibition is the process by which living or dead plant cells absorb water by surface attraction. Materials like cellulose or proteins are hydrophilic or water loving and they absorb moisture. Examples: dry seeds swell when soaked in water, wooden doors swell in rainy weather. It creates imbibitional pressure helping seed coats to burst during germination. Imbibition plays a role in ascent of sap in plants. Diffusion. Diffusion is the free movement of molecules like solute or solvent from a region of higher concentration to a lower concentration when it is in direct contact. Experimental example: add dye or potassium permanganate in water. These molecules move and spread evenly. Like diffusion starts progressing fully diffused and make the uniform solution. Key points include no barrier or force needed. Molecules move freely in all directions. Stirring speeds up the process of diffusion. Osmosis, it is the movement of water molecules from a region of higher water concentration dilute solution to lower water concentration or concentrated solution through a semi-permeable membrane. The membrane allow only water to pass, not the solutes. Important notes: osmosis here is about water, but other solvents can show osmosis too, specific to their membrane. Types of osmosis: endosmosis: water enters the cell when the outside solution is less concentrated than the cell sap, causing the cell to swell. Exosmosis: water leaves the cell when the outside solution is more concentrated than the cell sap, causing the cell to shrink. Experiment 2 demonstrating osmosis. Materials use thistle funnel, concentrated sugar solution, cellophane paper or egg membrane, beaker with water, and clamp stand. Procedure: fill the thistle funnel with concentrated sugar solution, cover the funnel's mouth with cellophane paper and tie it tightly. Invert the funnel into a beaker of water and clamp it in place. Mark the initial level of sugar solution in the funnel. This is called the experimental setup. The image showing the experimental setup here. Control setup: use another thistle funnel filled with plain water and place it in a beaker of water. This helps compare that changes happen due to sugar solution. Observation: in the experimental setup, the sugar solution level rises inside the funnel. In the control setup, the water level remains unchanged. So water in the beaker of the experimental setup is slightly drops but is not sweet. No, because no sugar has leaked there. Conclusions or observations from the experiment: water molecules moved from the beaker into the funnel through osmosis. Sugar molecules didn't move out into the beaker. Cellophane paper worked as a semi-permeable membrane, allowing only water, not sugar, to pass through. Experiment 3. If a more concentrated sugar solution is used inside the funnel and a dilute sugar solution or water is used in the beaker, water from the beaker still moves into the funnel and the level of solution in the funnel rises due to osmosis. This is the glass rod, this is thread, sucrose solution, this is the whisking tube, and this is the distilled water. What if we use rubber sheet or muslin cloth? Rubber sheet or muslin cloth is not semi-permeable membrane and water cannot pass through it, so no osmosis occurs and no rise in level happens in the funnel. Key concept reinforced: osmosis requires a semi-permeable membrane. Only water molecules move across it from low solute concentration to high solute concentration. Larger molecules like sugar cannot pass through. Osmosis demonstration using a whisking bag. Materials used: whisking bag, a synthetic semi-permeable membrane, concentrated sugar solution, capillary tube, beaker filled with water, and clamp stand. Procedure: fill in the whisking bag with concentrated sugar solution, tie the top of the bag around a capillary glass tube, and then immerse the bag in a beaker of pure water and support it upright. Make the initial level of the solution in the tube. Observation: after some time the level of sugar solution rises and enters the capillary tube. This happens because water from the beaker enters whisking bag thoroughly through osmosis. The rise is due to inward diffusion of water from the less concentrated pure water solution into more concentrated sugar solution inside the bag. Key points in the experiment: two liquids of different concentration. Beaker: water, more water molecules, fewer or no sugar molecules, more dilute. Whisking bag: more sugar molecules, fewer than water molecules, less dilute, and this difference in tonicity drives the movement of water. Roll of semi-permeable membrane. Cellophane paper or whisking bag acts as semi-permeable membrane. It allows water molecules to pass through freely. It blocks sugar molecules due to their larger size. How long does osmosis continue? Osmosis does on until both sides of the membrane have equal concentrations of water, that is isotonic condition. But in practice, water entering the funnel or bag increases the height of liquid. This creates pressure or hydrostatic pressure that opposes further osmosis. Eventually, the osmotic inflow stops when the forces are balanced. Now, concept is osmosis is water movement from high to low concentration across a semi-permeable membrane. The concentration difference and membrane properties control the process. It plays a vital role in absorption by roots and cellular activities in plants and animals. What is osmotic pressure? It is the minimum pressure that must be applied to a solution to stop the inward flow of water through a semi-permeable membrane. It's a measure of solution tendency to absorb water by means of osmosis. So, experiment demonstrating osmotic pressure. Now we find a thistle funnel with sugar solution is covered by cellophane paper and placed it in water. A piston with weight is placed on top. Result, the solution level does not rise, meaning water cannot enter. What is the conclusion? The weight or pressure applied is equal to the osmotic pressure. It prevents the process of osmosis. What is tonicity? Understanding the nature of solution. Tonicity refers to the relative concentration of solutes in two solutions and how this affects water movement. Three types of tonicity. Isotonic solution. Iso means same. So solute concentration is equal on both sides of the membrane. No net movement of water occurs in this process. So cells remain in the same size. What happens in case of hypotonic solution? Solution outside has lower solute concentration than inside the cell. So water enters the cell. It is endosmosis and cells may swell or burst. If too much water enters, plant cells become turgid. Hypertonic solution or hyper solution outside has higher solute concentration than inside the cell. Water leaves the cell or exosmosis and cells shrink. Plasmolysis in plant cells. Effect on different cells. In case of isotonic solution, cell shape and size remain unchanged. No net osmosis. Hypertonic solution, here cell shrinks as water exits. In animal cells become crinkled and in plant cells plasmolysis occurs. In hypotonic solution, cell swells may burst in animals. In plant cells it becomes turgid due to cell wall resistance. Active transport. The definition of active transport is the movement of substances against the concentration gradient from lower to higher concentration, and the process requires energy or ATP and leaving cell membrane. Example, uptake of mineral ions by root hairs from the soil, and opposite of diffusion, which is a passive process. This is the summary. And fate of animal cell in salt solution like isotonic solution, hypertonic solution or hypotonic solution. In case of isotonic solution, we have mentioned earlier, there is no change in shape or volume of the cell. So water entering the cell and is equal to the water leaving the cell. So the cells remain normal in case of isotonic solution. But if we put the cell in hypertonic solution, water leaves the cell more than it enters. So exosmosis occurs and the cell shrinks and becomes crenated or spiky. In case of hypotonic solution, water enters the cell more than it leaves, that is endosmosis, and the cell swells and may burst. This is called lysis. Fate of plant cells in salt solution. Plant cells, isotonic solution, no net movement of water. The cell remains flaccid, no gain or loss of water. Hypertonic solution, water leaves the cell, that is exosmosis, the cell sinks and the membrane pulls away from the wall, a process called plasmolysis. Hypotonic solution, water enters the cell, that is endosmosis, the cells become turgid as the vacuole swells and the cell wall prevents bursting here. Now we study active transport and passive transport. Active transport basically is the process where ATP is required and movement occurs from low to high concentration. It is seen in root cells absorbing mineral ions like nitrates, potassium, etc. against concentration gradient. These ions cannot pass easily across the cell membrane due to higher internal concentration. So they are actively absorbed using energy. Passive transport, there is no energy is required, ATP not used, and movement from high to low concentration occurs. It includes diffusion and osmosis. Passive transport is the free movement of molecules from their higher concentration to their lower concentration. Turgidity and flaccidity. Turgidity, when a plant cell takes up water and becomes swollen due to internal pressure. This is caused by osmosis. The turgid state is important for maintaining the rigidity and structure of plant cell. Turgor pressure, the pressure exerted by the cell contents, mainly vacuole, on the cell wall due to water intake. Wall pressure, the counter pressure exerted by the cell wall against the Turgor pressure. When the Turgor pressure is equal to wall pressure, the cell is in a balanced state. Water neither enters nor exits, maintaining cell stability. Bursting of cells, example fruits or vegetables, occurs when wall pressure fails to counteract the increasing Turgor pressure. Plasmolysis. Plasmolysis occurs when a plant cell loses water in a hypertonic solution and the plasma membrane pulls away from the cell wall. Flaccidity is the state when the cell is not rigid due to water loss. This is the difference between diffusion, osmosis and active transport. You can find it. Now We discuss the turgid state. When a plant cell is placed in fresh water or hypotonic solution, water enters the cell via osmosis. The vacuole swells and the plasma membrane presses tightly against the cell wall, making the cell fully distended and rigid. This condition is called turgidity, and turgid cells maintain structure and rigidity, especially in non-woody parts like leaves. Plasmolysis: when a plant cell is placed in hypertonic solution, that is example, for example 5% salt solution, water moves out of the cell. This causes the cytoplasm to shrink and plasma membrane to pull away from the cell wall. This process is called plasmolysis. The cell becomes flaccid, soft and limp. Plasmolysis is the reverse of turgidity. If a plasmolysed cell is placed back into pure water, water re-enters the cell, the protoplasm swells again, pressing against the cell wall. This reversal of plasmolysis is called deplasmolysis. The terms defined: turgidity means the rigid and stretched state of a cell due to water absorption and vacuole expansion; shrinkage of cytoplasm and plasma membrane detaching from cell wall due to water loss in hypertonic solution. Flaccidity: the loss and limp condition of a cell when water is lost, that is opposite of turgidity. Plasmolysis in practice: practical applications of plasmolysis: pickling, salting vegetables prevent bacterial growth via plasmolysis of bacterial cells; sprinkling salt can kill weeds by drawing water out of cells. However, excessive salting in agricultural fields can damage root cells and reduce crop yield due to plasmolysis. This is the image showing a turgid cell. The cell after immersing a hypertonic solution, showing the successive stages in the shrinkage of the protoplasm from the cell wall caused by a withdrawal of water from the vacuole, and after immersing the cell in a hypertonic solution. Use of turgidity to plants: provide rigidity to soft tissues like leaves. When there is a less water, leaves wilt, petiole and lamina lose firmness. Turgidity keeps leaves upright and spread out, maximizing photosynthesis. Helps plant resist drooping in hot, dry conditions by maintaining structure. In the evening, when transpiration is reduced, turgidity restores leaf shape. And turgor pressure helps the push through hard ground. Example, in mushroom and germinating seeds, turgor pressure enables the structure to push through the hard soil layers. Here the figure shows a germinating seed developing force back to the soil. Even concentrate floors or walls can crack due to higher turgor pressure in plant roots. Build up root pressure: here the root pressure is the upward pressure created in roots due to water intake by osmosis. When a well watered plant like balsam is cut and connected to a glass tube, water rises due to internal pressure from the roots. Demonstration with a manometer shows mercury level rising as water, that is cell sap, is forced upward by cell root pressure. Root pressure is the pressure developed in the roots due to continued inward movement of water through cell to cell osmosis, which helps in the ascent of cell sap upward through the stem. Turgor in the opening and closing of stomata. Guard cells regulate the opening and closing of stomata. These cells become turgid when they absorb water, causing them to arch outward, opening the stomatal pore. At night or underwater shortage, guard cells lose turgidity and become flaccid, causing closure of stomata. This movement is crucial for transpiration, gas exchange and water conservation. Turgor movement in sensitive plants like Mimosa pudica. It's a classical example of turgor based movement. Touch stimulus causes a rapid loss of turgor in the pulvinous tissue, a swelling at the leaf base, and leaf flakes folds within 2 to 3 seconds, then gradually reopen. The pulvinus at the base of the petioles in plants like Mimosa pudica loses turgor when touched, leading to drooping. After some time, turgidity returns and the leaves stand erect again. Similar turgor movements occur in insectivorous plants that snap shut to trap prey. Sun tracking flowers and slip movement of leaves all are driven by turgor changes in specific cells. Imbibition and turgor together generate force. Imbibition is the passive absorption of water by dry substances like cellulose and starch. Seeds and grains swell when soaked because imbibition pulls in water, and turgor pressure builds up inside the cell due to osmosis. This is internal pressure can burst seed coats and crack walls of grain storage rooms. Example: soaked seed burst containers due to internal pressure. Rainwater causes basement crack in grain storage due to swelling seeds. Root pressure. The definition: when you cut the shoot of a well-watered plant, water extrudes from the cut end due to root pressure. Root pressure is caused by cell-to-cell osmosis in the root cortex mechanism. Water enters root hairs by osmosis. Water moves through epidermis into cortex into endodermis and pericycle. Water finally reaches the xylem. Hydrostatic pressure builds and forces water upward through the stem. Guttation. In plants like tomato, grass, banana and ferns, root pressure is strong enough to push water into the leaf tips. This excess water is lost as liquid droplets on leaf edges, mostly in early morning. This process is known as guttation, not be confused with transpiration, because which is water vapor loss. Importance of root hair and upward water movement. Function of root hair: root hairs absorb water from the soil via osmosis. The image showing the process. The cell sap inside the root hair is more concentrated than the surrounding soil water. Once water enters the root hair, it travels cell to cell to the xylem vessels, and this continues until water enters the vascular tissue and is carried upward. Water absorption and upward movement. Water is absorbed by root hairs through osmosis from the surrounding soil. The process is aided by root pressure generated by osmotic flow, Turgor pressure in root cells, active transport of minerals as ions. Once absorbed, water travels from cell to cell, eventually reaching the xylem vessels. From the xylem, water moves upward to the stem and leaves. Experiment on absorption and conduction. Experiment 1 is to show that roots absorb water. Set up test tube A is the control test tube filled with water and oil layer to prevent evaporation, and test tube B is the experimental one, which contains a plant with roots submerged in water also covered with oil. Observation: after 1 to 2 days, the water level in B decreases, proving that roots absorb water. Water level in A remains unchanged as no plant is present to absorb water. The image showing that roots absorb the water. Experiment to show that water is conducted through xylem. Set up: a balsam plant is placed in a beaker containing eosin solution, a pink dye. After 3 to 4 hours, the plant is removed and washed. Observation: transverse section of the leaf and root show pink staining only in the xylem tissues, and this proves that water travels only through the xylem vessels. Vascular bundles in plant: the xylem and phloem form a continuous system of tubes. Xylem transport water and minerals upward, we know that, and phloem transport food substances like sugars up and down the plant. Conduction of water through xylem. Set up: take a leafy shoots or balsam plant, place them in water after cutting. For beaker A, it remove a ring of phloem only, xylem remains there, and for beaker B, remove a ring of xylem only, phloem remains. Observation: in beaker A, the twig remains turgid and healthy, proving water is being transported via xylem. In beaker B, the leaves wilt and droop due to lack of water conduction, showing xylem is essential for upward water transport. The image showing water is conducted upwards through xylem. Experiment 4: conduction of food through phloem. Set up: select a woody stem, or example guava, and remove a ring of bark deep enough to cut through the phloem but not the xylem. It's called girdling. Let the plant remain for several weeks. Observations: above the girdled ring, the stem swells due to accumulated food. Below the girdle, the stem stops growing and may even decay or die due to lack of food from the leaves. This proves that phloem conducts food downward from the leaves to roots. Forces contribute to ascent of sap. Root pressure: it builds internal pressure in the xylem due to water uptake by roots via osmosis. This pushes sap upward in the xylem. More effective in herbaceous plants but not sufficient for tall trees. Capillary or capillary action: water rises in narrow xylem vessels due to capillary force. The narrower the tube, the higher the water rises. It helps fills the vacuum created by transpiration. Transpiration pool: the main force in all tall trees. As water evaporates from the leaf surface, water molecules are pulled up to replace them based on cohesion, that is, water molecules stick to each other and forms a continuous water column from roots to leaves. Addition: water molecules stick to waters of xylem vessels. This helps prevent the water column from breaking. Addition also pulls water upward as molecules cling to the walls and draw others behind them. Water up and down, molecules by molecule. How does water reach the top of a tall coconut tree? Water is absorbed from the soil by the roots as transpiration occurs in the leaves, that is, water evaporates from the stomata as a suction force is created. This causes another water molecule from below to rise and take in place, and this chain of molecules continues in succession from the roots upward through the stem and leaf by leaf. The entire water column remains continuous and unbroken due to the force of cohesion. Cohesion is the molecular attraction by which the particles of a body are united throughout the process, throughout the mass. Can you guess from where the sweet taste of the coconut milk comes? While water reaches the fruit via xylem, the sweetness of coconut milk comes from food substances, mainly sugars, transported by the phloem from the leaves during photosynthesis. This is the chapter summary. So my dear students, roots are the plant's hidden helpers. They absorb water and minerals from the soil and send them up to the leaves with the help of forces like osmosis, capillary action and transpiration pull. This keeps the plant healthy and helps it make food. Thanks for watching. If you enjoyed the lesson or chapter, don't forget to like, share and subscribe my channel for more such fun biology classes. Stay tuned, stay rooted in learning. See you in the next time. Bye.